Our next speaker is Professor Raquel Peixoto, and she will be talking to us about robotic customized medicine for corals.
I think you all be quick, especially because luckily Adrienne already introduced the topic for me, so I can skip lots of slides. And I will talk a little bit about probiotics, which is something my group has been developing. Probiotics for corals, we've been developing this for quite a while, and solutions and ideas for these application in the field in a more automated way, but where we came there. So I will skip the explanation about why corals are in danger. I think Adrienne already did a great job on explaining why microbes are important. And in our case, we are very focused on the bacteria and also some of the other microbes, but specifically bacteria. And I think it's also very important to bring some of the basic concepts that actually inspired us to develop this type of approach. So I'm a microbiologist, and for all of us, for all living organisms, microbes are very important. We all rely on the microbes living within us. And but this is the good and for the bad as well. So if everything goes well, our microbiome is very important for us. It supports us, it gives us vitamins, it helps us metabolize some of the compounds you need. But also if we somehow, for some reason, change these balance and change the microbiome that's living within us, they can also cause disease. So microbiome stewardship is something that is being implemented for humans as well to treat disease. So basically what we need to do is that we need to try to always keep our microbiome, our original microbiome, our beneficial microbiome in shape. And we can do that through what we eat, through our behavior, to what we are exposed to. And for us, this is a little bit not easy, but it's a little bit more straightforward because we know more or less what to avoid and how to do that. But for corals, it's a little bit more difficult because they cannot move. They cannot go somewhere else that is colder or it's nicer, but we can do this for them. So the basic concept here, when we think about microbiome stewardship, is that when we or any other organisms, including corals, are exposed to stress and impacts, what usually happens is that the beneficial microbiome is replaced by pathogens, because most of these pathogens are opportunistic, and they actually take place when the microbiome, the original microbiome, the native microbiome is hit, it's impacted. And some of them, especially for corals, are activated by heat. And this is a very basic ecological concept, right? And it's very important to highlight as well that when we think about microbiome stewardship, we are not really, because very often we say that we need to manipulate the microbiome of corals, that we want to manipulate it. But in fact, we are not manipulating anything, because the microbiome has already been manipulated by the impacts that we are exposing these corals to. And this is the same for us when we are sick. And so the basic concept is that there are no pristine ecosystems anymore, and there are no pristine microbiomes anymore, especially when we talk about corals and coral reefs. So when we think about microbiome stewardship, we are actually trying to retain the good bacteria, to restore the good bacteria, to rehabilitate it in some cases. So the very basic concept here is that impacted corals will have the microbiome being changed towards something more pathogenic. So the beneficial ones, the original ones that were there, they are very common, very abundant in the ocean. They are replaced by pathogens. And these pathogens, they overgrow the beneficial bacteria and they can spread. So we have an additional problem that we have to think about, that in the future, the damaged reefs can become sources of pathogens that will actually also impact other organisms. And if you think about that One Health concept, it can also reach us. But this is a talk for another time. And basically what we are trying to do here, as we revegetate forests that are degraded, we are trying to restore microbiomes that are degraded. And we do that for humans as well. This is not a crazy idea. It's being used for plants and humans for decades. It started to be used also for wildlife, bats, bees, frogs. They're all being treated with probiotics with very good results. For corals, more specifically, when we started working on this, we went to the literature because I actually started my career as a plant microbiologist, and it's very straightforward for plants and agricultural fields. We have a framework to use probiotics. We know the beneficial roles. We just go there, we isolate bacteria, we look for these beneficial roles, we assemble them, and we apply back. When I started working on corals, I went to the literature, and there was nothing there. Right, there was a very nice paper on the coral probiotic hypothesis indicating that the microbiome associated with corals should be beneficial, which is something we know for all organisms, but there were no a framework. And I was, of course, inspired by these and also the assisted evolution approaches that people were suggesting, but there were no actually mechanisms. There was no mechanism for this to be developed. So I went to the literature, I started to think about the coral physiology as well. And we started to think about mechanisms, specific mechanisms that could be a parallel to what was being done for humans and plants, but that could be beneficial specifically for corals. And as a microbiologist, I went through a list of things that were beneficial for other organisms, tried to think if that was also the case for corals, included a few things. For example, we know that reactive oxygen species is one of the problems when corals start to get exposed to heat stress. It's toxic. It also causes problems to both corals and the algae that is associated with them, the symbiodiniaceae that Adrian also talked about, DMSP, which is also a compound that is common within the holobiont, but when corals are stressed, it increases in concentration and it attracts pathogens. This was also something that we thought, well, bacteria can degrade them. Bacteria can mitigate ROS as well, so let's use them. And the idea here was to combine these mechanisms into a consortium. So we had more than one of these mechanisms. We could apply them back. And these are original bacteria. These are bacteria that we isolate from corals. They are good, but they are sensitive. So we basically isolate them in the lab. We assemble them, considering the beneficial mechanisms that we think that could help corals go through times of stress. We put them all together so we can tackle several of the different problems, a cascade of problems that are actually triggered by heat stress or any other impact. We apply them back. And when we tested this concept, we've basically managed to see this in the lab. We proved the concept that we could combine these mechanisms and apply them to corals to mitigate heat stress. We went forward. We could see in another experiment when we increased even more the impact that these probiotics could actually protect corals against mortality. All of these in the lab. And we went on, and other people also went on, and we tested different coral species, different coral combinations of probiotics, different goals. We wanted to see whether they could protect corals against disease, against and other people as well. We wanted to make corals grow faster. We also had transplantation of the microbiome being published, and in all of these cases, we had beneficial effects and protection through the use of microbiome. And this is also very complementary to what David presented this morning, because in this case, we want to protect the corals that are already there in the reef, right? So we need to protect these corals to retain these colonies and the genetic maturity and diversity that we have in the reef, in parallel of developing restoration approaches that can actually rebuild the corals that are already damaged. And it can also be used for the nurseries, because one of the limitations is time, that these corals don't grow very fast, and we can use probiotics to make them grow faster, because they can accelerate classification, they can make them healthier, they can increase the amount of nutrients, so we can also use these in nurseries. And that said, crash course on coral probiotics, that we went all the way on testing different conditions, different probiotics, different coral species. And every time we had good results, we decided that it was time to go and test it in the real world to see, first of all, if this concept could be translated, that meaning whether we could actually deliver probiotics to corals. And we are also testing other ecological aspects on whether these probiotics can also influence the microbiome and the health of other organisms in the reef, because of course we want to protect the corals, because if we protect the corals, we can protect the coral ecosystem, as they are foundation species, but we want to see whether there is a direct influence and effect of the probiotics on the other organisms as well. And to do so, we spent some time building the right ecosystem, the right laboratory to test this. And we established the Coral Probiotics Village quite close to KAUST, 20 minutes from our marina. And basically, this place was selected because we wanted this place to be somehow protected. We wanted this to be shallow. We wanted several patches of reefs to be available. So we have these patches that we use as mini reefs, as replicates. I show you the images while I talk about it. We wanted some specific species to be there. We wanted to be able to monitor it very closely. So our group goes to the reef every week, sometimes 3 times a week. We have probes and we have some equipment in the reef to monitor it over time, all the time. And we have data that is actually being collected to correlate with the data that we are collecting. We have other PIs that are doing the research in the same area. So by studying the exact same area, we can correlate our data. And we also have colleagues testing their toys as Shinkyu in our village, because, and also Professor Charlotte and Manuel and Carlos, Sebastian, all of the people coming around either in the village or in the neighborhood, because then we have a lot of data that we can exchange in the future. And the city started because when we started inoculating the corals, and you saw me with the syringe, because so far this is a very well-controlled experiment. So we are inoculating specific colonies and monitoring specific colonies in the surroundings of these colonies. We are divers, and there is a time constraint in this case because we needed to find the next colony and the next colony. And this is why we started to tag these colonies and connect them with ropes, because we could easily find the next colony that needed to be inoculated. And we also started to mark these areas with street names. So Nemo Street, for example, is an area that we know that is full of anemones and clownfish. And so that made us it made it easier for us to localize ourselves underwater. And this is why it became a city, and is also a great opportunity for outreach. The kids at school love it, and the community as well. And we are monitoring the physiological states of these corals very closely. So we are using some equipment that we can use underwater, and we are collecting in situ and real-time data on the health of these corals, but also the sponges, the algae, and other organisms in the reef, including fish and the microbiome, to see how it's influenced by the inoculation of probiotics. So we have 2 main goals here. We have the overtime monitoring of the connectivity, the microbial connectivity in the reef. So we are testing, we are collecting corals, sponges, algae, the same individuals over time, water, sediment, fish, and we want to see how the microbiome of these corals change over time in a natural environment, and we correlate this with temperature, salinity, all of the data that we are collecting. But we are also monitoring the patches that are being, and these are the patches being inoculated with placebo. So we are not doing anything, and we want to see how it naturally happens in the reef. And in parallel, we are collecting the data from the patches where we are inoculating probiotics. And in all of these cases, we are collecting physiological data. So we want to see how the microbiome changes over time, whether these microbiomes are connected, and we want to see how this is associated with the health status of these organisms. And we are also checking whether the inoculation of probiotics can influence these traits. And because it's very complicated what we are doing now is that we're trying to prove the concept that this can be used in the reef, and also checking risk assessment and ecological outcomes. And this needs to be done in a very well-controlled way in specific colonies, but we are already thinking of the future. As we know, we have a very narrow window, so we are developing several things at the same time. And one thing that we are developing here in KAUST in collaboration with Professor Khaled Salama's team is that we are developing an underwater irrigation system that can deploy the probiotics in an automated way. So basically, we have containers where we put the probiotics, and in parallel, we are developing in the lab ways to produce these probiotics so that they can stay alive for a long period of time. We put these probiotics inside the container, and we spread it with hoses that go, these hoses go around the reef, and it can actually work exactly as an irrigation system that we have in the backyard of our houses here at KAUST. And the idea is that in these cases, very located because we are inoculating specific colonies, but the idea is that it could be in the entire reef, and it could be inoculating probiotics, so divers would not need to go and re-inoculate it. And this is the funny slide that I presented when I came to KAUST. It was like during my presentation of how I envisioned the application of probiotics, and I had this idea that we could have a shower or something. And this is actually what we are developing with Khaled, and I think it's very close to that. And again, this is very specific for this colony, but the idea is that we can have it exactly as we have in our backyard. It's not in every single patch of grass, but it can cover a specific area of the reef. And the good news is that back, it's not coming back. Okay, the good news, the good news, they don't want me to share the good news. The good news is that our preliminary data that we've collected so far in one of the experiments that actually, this one we were using the syringe, indicate that yes, we can change the microbiome of corals in the reef, in the real world, because the first question is whether this is going to be washed out. But the first results indicate that some of the bacteria we're inoculating are being incorporated by the corals in the reef. And I think it's an even better news that the water and the sediment, the microbiome of the water and the sediment that is just around the coral, it's the same. It's not being changed by the inoculation of probiotics. So it's quite specific. And we are isolating these corals from the corals, these bacteria from the corals, which makes sense. We are developing some other tools to monitor the health of these corals, also together with Professor Salama, trying to develop microneedles that can help us monitor the microbiome and the physiological responses of these corals over time, also in an automated way in the reef, but this is still very preliminary. And because I don't have much time and I try to actually go super quickly, I will go straight to the team. Oh my goodness, this is not... because these are actually, it's a lot of people to develop all of these things. These experimentalists, this is the people that are actually still working with me in Brazil. Some of them are already here at KAUST, so this is not very updated. Some are here at KAUST, but it takes the entire team to work at least for 10 days, every day, the entire day, for each sampling time of that massive experiment that we are running in the reef. So these are the real stars behind all of the data. I'm just the person asking the crazy things to be done. I go with them as much as I can, but they carry the heavy lift. And with that, I thank you. And I hope that I've managed to kind of catch up with the agenda. Thank you very much.